Hello and welcome to Nikolay's genetics lesson. This video going to be about population genetics, and here's a problem. Below are the genotypes at 3 loci for a sample of 6 individuals. What would genotype frequencies be at locus 2 in this population if it were in Hardy-Weinberg equilibrium? And as usual, I recommend you to stop video here, try to solve this problem on your own first, and when you would be ready you can run video again and you can compare your answer with my answer and explanation. Because we are told that we have to find frequency of the locus 2 in the F1 generation, we can ignore information for the locus 1, so we can cross out it, and locus 3. So let's just cross out these two loci, and here is the Hardy-Weinberg formula. So frequency would equal two alleles. We see here two alleles. One is recessive allele E, another is dominant allele E present here. So frequencies of the three genotypes that can be made by two alleles would equal to p squared plus 2pq plus q squared, and would equal to 1. And p squared here stands for the homozygous dominant genotype, so capital E, capital E. 2pq stands for the heterozygous genotype, capital E, small e. And q squared stands for the homozygous recessive genotype, or small e, small e. And all the alleles p plus all the alleles q, when we add up these frequencies, would equal to 1, or 100%. So 1 in our calculations equal to 100%. And P, as you already saw, equals to capital E, or dominant allele E, and Q is recessive allele E in our calculations. So now we have to find frequencies of the P and Q allele, so we can use these frequencies in our calculations, in our formula. So let's do it. And here we see 12 alleles, and recessive allele E here present in 2 copies. So frequency of the recessive allele E would equal to 2 divided by 12. So 2 recessive allele E divided by total number of alleles. And our answer going to be 1/6, and decimal number would be 0.17. This is rounded number. And now we can put this frequency here. So we have found the frequency of the recessive allele E, or Q, and instantly we would be able to tell the frequency of the dominant allele P. This is going to be 0.83. When we add these two numbers, we are going to get 1. So now we can find frequency of the homozygous recessive genotype, that is q squared. So q squared, that is going to be frequency of the homozygous recessive genotype, would equal to frequency of the allele Q, or recessive allele E, that is 0.17, and squared. So 0.17 squared. And our answer here would be 0.03. This is also a rounded number. And we can put this frequency here, 0.03. And now we can find frequency of the heterozygous genotype, that is 2pq and capital E and small e. So this would equal to 2 multiplied by the frequency of the allele P, that is 0.83, and by the frequency of the recessive allele E, that is 0.17, or recessive allele in our formula is Q. So our answer here would be 0.28. This is also rounded number. We can put this frequency in our formula, 0.28. Now we can also tell the frequency of the p squared or homozygous dominant genotype just by adding these two numbers and subtracting from 1. But let's also calculate this number just squaring frequency of the allele p or dominant allele e. So this is going to be p squared, and genotype would be capital E, or homozygous dominant. So 0.83 squared would equal to 0.69. So let's put this frequency also here, 0.69. And when we add all these three numbers, we are going to get 1. So our calculations are correct, and this is going to be frequency of the three genotypes in F1 generation. So this is going to be our three answers for three genotypes. And also my comments would be that Hardy-Weinberg formula works for very large populations that where individuals made randomly, where there is no mutation and no migration. And as you see in our example, we have parental generation that has only 6 individuals. So this formula would work if this would be not animals that produce very small number of progeny, but such animals like fish, for example, that can produce millions of fish eggs and millions of offspring, then this formula would work fine. And this formula would also work fine if our initial population also would be counted in hundreds and thousands of individuals that would produce small number of offspring. So you just have to know it when you do such calculations, that size of population matters, and you have to assume that there is no migration that can change allelic frequencies when some of the representative of this population can live, or other group of animals can join this population and so can change allelic frequency in our population. So in Hardy-Weinberg formula, we assume that there is no migration, no new alleles appear due to mutation. There is no assortative mating. Mating is random. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.